Hello, my name is Tim Robinson, and today the scientific paper that I'm going to be reviewing is about CRISPR-Cas9 and how we can use this technology to edit the genome of crops, improving them. I'm a biology teacher, and we talk about genetics, and this is the tip of the spear of genetics. And what we're going to talk about in this is going to be really paving the way of how science can improve our crops in the future. This is a revolutionary technology that will improve the crops that we feed the world. For centuries, humans have been modifying plants through traditional breeding, a process of careful selection and crossing. While this is effective, this method can be slow, which often brings along unwanted traits. More recent techniques like chemical and radiation mutagenesis introduce random changes in the genetic code, requiring extensive screening to find beneficial mutations. Now, the limitation of these older methods paved the way for this new era of this thing we call site-directed mutagenesis. This approach uses engineered molecular tools known as site-specific nucleases, or SSNs for short, to make precise changes in a plant's DNA at specific locations. The review we're focusing on today is highlights the journey towards this precision, mentioning earlier tools like mega nucleases, zinc finger nucleases, and transcription activator like effector nucleases, or TALENs. These were significant advances, allowing scientists to target specific genes for modification. However, the CRISPR-Cas9 system is the main focus of this review. Like, we've done other things to edit the genes, but this is the most revolutionary part that we've discovered in the last 20 years. So this is a picture of a Cas9 molecule. At its core, it is a two-component system. First, there is a Cas9 protein, which is the yellow part in there, which is an enzyme which acts as a pair of scissors, or molecular scissors, capable of cutting or splicing the DNA molecule. Second, there is the gRNA molecule, the gray part right there, which is the guide RNA. This is a short RNA molecule designed to be complementary to a specific DNA sequence in the plant's genome, meaning that it matches a specific DNA sequence or a genome that we want to target. Think of this as like your GPS that directs the Cas9 scissors, so it guides it to the exact location we want to edit. So how does this kind of work? So this picture right here is a perfect image of what we're looking at. The beauty of the Cas9 system lies in its simplicity and adaptability. The gRNA guides the Cas9 protein to the targeted sequence. Once there, the Cas9 creates a double strand break in the DNA, essentially cutting it out. The plant's natural DNA repair mechanisms kick in. Scientists can exploit these repair pathways in two ways. Non-homologous end jointing, or NHEJ, which often leads to small insertions or deletions, effectively knocking out a gene, or homology-directed repair, HDR, which in the presence of a DNA template, which we can program, allows for a precise gene insertion or replacement. So if we don't like a gene, we can cut it out, or if we want to change a gene or add a gene, we can put in whatever we want. This review emphasizes that CRISPR-Cas9 is far more than just a gene editing tool. Its versatility is constantly expanding. Researchers are developing techniques for multiplex gene engineering, allowing for simultaneous modification of multiple genes all at the same time. Furthermore, CRISPR can be adapted for gene activation or repression, controlling how much of a gene is expressed without permanently altering the DNA sequence. This opens up exciting possibilities for fine tuning plant traits. Now, in our next slide, these are the different ways, or one of the key applications highlighted in this review is the nutritional improvement. Imagine crops engineered to have higher levels of essential vitamins, minerals, healthier oils. CRISPR-Cas9 makes this way more achievable relatively. By precisely modifying genes involved in the nutrient production or accumulation, we can develop biofortified crops that allow for that address our malnutrition and improve human health overall. Also, this review also delves into the crucial area of enhancing plant resilience. With climate change, we will start to see lots of growing zones start to change around the world. Disease and environmental stresses like drought pose a significant threat to crop yields. CRISPR-Cas9 offers powerful tools to engineer plants with enhanced resistance to specific pathogens or increased tolerance to water scarcity by modifying the genes involved in plant defense mechanisms or stress response pathways. We can develop more robust and reliable crops by editing them. So the next, a critical aspect of gene editing is how the CRISPR-Cas9 components are delivered into the plant cells. Traditional methods often involve using these things called plasmids, or these circular DNA molecules, just how bacteria also transfer their DNA to one another. To carry the Cas9 gene, which carry the Cas9 gene and the gRNA together. However, this approach can sometimes lead to the integration of other foreign DNA into the plant genome. This review highlights the excited development of CRISPR ribonucleoproteins, or RNPs. These are pre-assembled complexes of Cas9 protein and gRNA, which can be directly delivered into the cells, offering a potential cleaner, more transient way to perform gene editing without the risk of stable transgene integration, meaning giving DNA that we do not want inside of our plant cells. In conclusion, this review clearly positions CRISPR-Cas9 as a groundbreaking innovation in plant biology. Its rapid evolution, its versatility, and diverse applications ranging from nutritional enhancement to improved stress tolerance hold immense promise for the future of crop improvement and global food security. As the technology advances, we can expect even more sophisticated and targeted approaches to create the next generation of crops. This technology is powerful and amazing. And if we can use it for our crops, we can change the game on food insecurity. And it's an amazing technology, and I love it. And I love talking about it because I'm a biology teacher. All right, so that's all I have for you guys today. Thank you very much. If you have any questions, make sure you leave it in the comments. I highly suggest you look into this tip of the spear gene editing stuff. it is going to be the future as we go into either into space or as climate change starts to have its effects. This could be the the next big thing, or it is the next big thing. All right, I'm done. Thank you very much. Have a good day.